Hi there, I'm Dennis, and I'd like to thank the organizers of the conference for inviting me today to talk about applications and challenges of machine learning and artificial intelligence in pharmacology. I'm speaking to you today from the University of Sheffield and the A-STAR Singapore Institute for Clinical Sciences. So when do we need machine learning? Well, what's great about computers is that they can process vast amounts of data in a short few moments, whereas humans can only process limited amounts of data through things such as our eyes or ears. So this creates an opportunity for using algorithms to derive new insights from large or vast amounts of data. But not to be outdone, humans can still learn very complex tasks with limited amounts of information. So how can machine learning accelerate drug development? Well, we know that the drug development pipeline can take very long and actually take up a lot of resources. So here today, I'll talk about kind of three instances where machine learning can help us identify drug targets, predict dose response of drugs, and also stratify patients at various stages of the drug development pipeline, thereby increasing the chance of success for a drug trial and also reducing its cost. So the application of machine learning techniques in translational research or drug development can take a few steps. Generally, it involves collecting large amounts of high-dimensional data, say from a patient's clinical records, from their genomes, proteomes, etc. And that data needs to be processed and made machine readable, and then also identify the relevant features in the data that can be used by algorithm to do generally one of two tasks. One of them could be clustering or grouping patients or samples based on similarities and differences. The other could be labeling the samples based on specific characteristics like patients that respond to a drug or respond to no drugs. So I'll give you first an example of how we can use kind of data-driven techniques and machine learning to identify new targets. And this involves kind of class of models known as the network-based models. So a lot of the kind of early advances in this field has been done in cancer research. And our experience with Microsoft Research was to develop a network-based model of an acute myeloid leukemia cell using reverse phase protein array data and information from published literature about signaling pathways that are relevant in this disease. And the network model performs two tasks of predicting the cell's fate, such as proliferation, the cells growing, or apoptosis when the cells die. And what's unique about these types of models, or what's useful about these types of models, is that we can describe all the different interactions in the components of this model. So essentially, the signaling pathways that drive these cellular processes. And you can perturb these processes. For instance, you can say, hypothesize that there's a drug that targets a particular component in this network, like a protein in this network, and then observe how the cell's fate might change computationally, how the cells might change given a perturbation by a potential drug. So this is very useful for testing different kind of target hypotheses, and it has been extended in more recently in COVID research, whereby scientists like at MIT have collected different types of data like RNA expression data, protein-protein interaction data, drug target data from lung cells infected and uninfected by the SARS-CoV-2 virus. And what the aim of this approach is to use a class of deep learning techniques called autoencoders to identify drug signatures that can reverse the expression patterns that you see in infected SARS-CoV-2 cells. So the idea is like if there are drugs where there are that has an effect in other diseases, whereby you can use that effect on SARS-CoV-2 to reverse the changes you see in the lung cells, then those drugs might be effective for treating COVID-19. Okay. And what's also unique about this kind of approach is that they not only identify the drug signatures, but they also map the drug targets, potential drug targets, to the genes that are differentially expressed in the infected cells. So then this model would also propose a causal mechanism for how a drug might change the expression of the genes in infected cells. Another example I'm sure you've come across is what Google DeepMind have been doing in predicting the structure of proteins. Now, it's important to predict the structure of proteins because we can then design better drugs that can bind to specific protein targets. And experimentally, determining the protein structure can be very costly. So it's important to have good computational techniques that can predict the protein structures, and in particular protein folds. So DeepMind had developed an algorithm called AlphaFold for doing this, which was quite successful in kind of out competing other methods in more accurately predicting protein structure. And the method really requires data from protein structures. Okay, so there's a kind of structural database, protein structural database that the algorithms learn from. And also it's augmented or supplemented by a genetic database. So these are kind of genetic profiles of not just humans, but different organisms. And using this genetic sequence, we can translate that into potential pseudo kind of protein structures that the algorithm can also learn from. Okay? And the secret sauce of this algorithm is that there's kind of two kind of deep learning modules that kind of integrates this kind of genetic information and the experimental protein information to help the algorithms learn and predict the protein structures. Okay? And just to give you a glimpse of the size of these data sets, there's basically 184,000 experimental structures that the algorithms learn from, but there's over 2 billion sequences that they can use to create new protein structures that the algorithm can also learn from as well. So there's actually quite a huge amount of data processing and bioinformatics to kind of create these augmented data sets that can be used to train these powerful algorithms. So some of the challenges of applying machine learning to kind of target discovery is that there's usually requires kind of a high level of domain knowledge in terms of the biology around the cellular system and, say, the proteins and pathways involved in disease. There's often need for a lot of manual data processing, as in the case of building the network models, and also a lot of the models are very kind of disease specific, so they're not as easily generalizable to other diseases or biological contexts. So the next example I'll give you is kind of when we have a few kind of investigational compounds and we're trying to predict the response of these compounds. Okay? And the approach we've taken before is try to extract as much information as we can from the drugs themselves, the chemistry and so forth, and also kind of the cells that we're targeting or the tissues we're targeting. So we can extract different like molecular or genomic data and kind of input and integrate these features in a machine learning model. Some of them could be like using deep learning techniques. Some could be more traditional machine learning models. And the task is to kind of predict the drug efficacy or toxicity. What I'm showing here is like kind of the efficacy of a combination of drugs as measured by their synergy score. And we often try to kind of come up with a confidence in that prediction as well. So an example of this is work being done by a talented PhD student, Tao Pham, who has used chemical information from pairs of drugs to predict the drug-drug interaction of those two pairs of drugs. And this is taking the kind of the chemical structure of those drugs and transforming and encoding them in kind of numeric vectors that can be used in a deep neural network to then predict essentially three types of labels, whether there's no significant drug-drug interaction, potential interaction, or severe kind of possibility of interaction. And the data that that's being used to train the algorithms come from the Liverpool Drug Interaction Database that classified different pairs of drugs into these three categories. And so then what we do is just essentially try to measure the performance of classifying pairs of drugs into these three categories correctly. So a lot of times you use classification metrics like accuracy, specificity, sensitivity, and we want to test over many different combinations of drugs. And as you can see, the performance is not always consistent, right? So for different labels, you might do better, and some kind of classes of drug-drug interactions, you might actually do worse. Okay? And it kind of depends also on how many of that class you have in your training data set. So you can imagine if you have less kind of red classes or those drug-drug interactions that are severe, then your algorithm is not going to be learning as well if there are less examples of those. So the other thing to consider is that there's not just use one algorithm that you can use. There's also many different machine learning algorithms that you can use, and actually many different ways of processing the data for these learning algorithms as well. So you want to try different kind of approaches, machine learning approaches, but you want to try them in an unbiased manner. So what we often do is split the data into a training set and a test set, and train multiple machine learning approaches on the training set, and then score and benchmark those approaches on the test set. And this is a technique very often used in kind of machine learning competitions where they're trying to benchmark, what is the best solution, such as the one that was submitted for AlphaFold. So this is a particular kind of benchmarking study we did on machine learning algorithms to predict the efficacy of pairwise drug combinations. And there's kind of 76 machine learning algorithms that were used. And you can see there's a variety of kind of prediction accuracy. So here we measure prediction accuracy by using the Pearson correlation between the observed and predicted synergy scores of the drug combination. And what you need to what's difficult to describe is how good is the predictions or how poor are the predictions. So what we use is kind of random models and also replicate experiments to kind of benchmark or lower bound or upper bound these predictions. And what you can observe here is that actually quite a lot of algorithms actually do quite well on the level of kind of replicate experiments as well, okay, for predicting drug synergy. But when we look at the kind of the predictions a bit more deeply, we can see that actually there are certain drugs that are predicted well, but there are also many drug combinations that are all consistently predicted poorly, no matter what machine learning algorithm is used. Okay. So there are characteristics of certain drugs that may just be difficult to learn from. And when we looked into those cases a bit more deeply, we found that actually the data we're training on is somewhat uncertain for those drugs. For instance, when you're training on dose response, so for instance, dose response statistics like IC50s that are outside of the viable concentration ranges, there's actually more uncertainty in the response of the drug, which is sensible, right? So what we try to do is like we need to use additional machine learning techniques to try to, or probabilistic techniques, more specifically, to quantify the uncertainty in actually the data we're training on, and actually include that uncertainty in the machine learning models. This is very often not done, but it's very important to actually tell the machine learning algorithms what data sets are actually, or data points, there's more uncertainty, and what data points there's more certainty. Okay, so just three kind of main challenges with the kind of these applications of machine learning to kind of predicting drug response data. You often have high dimensional data. So there's not only kind of chemical information chemical structures to consider, right? There's kind of bioactivity and kind of molecular profiles of the cells or the genes you're targeting. So you actually have quite a lot of high dimensional data, and this creates a problem of a curse of high dimensionality, where you have actually more features to consider than there are actually samples to train on. There are also many different data types that you need to consider, right? From chemical information, from biological information, from even clinical information. And then lastly, which I alluded to, is that there's some uncertainty in some of the data sets. Not all the data sets or the training labels are very certain. So you need to account for that. Okay, the third example is more on kind of application on patients specifically, and these kind of machine learning or data-driven approaches have been mainly driven by our ability to collect large amounts of data directly from the patients. Okay, an example of this, again, more recent work on COVID, is the ability to kind of collect samples from COVID patients and sequence the viral samples, and also the blood of the patients. Right? So this creates vast amounts of data, which can inform the evolution of the SARS-CoV-2 virus throughout the pandemic, and we use classification algorithms to identify new variants in the SARS-CoV-2 virus that appear over time, right? And I think these algorithms really reduce coupled with high throughput sequencing, we reduce the time to learn from large population data sets. In kind of the HIV field, another example of applications of machine learning techniques is And I think we're quite familiar with it in COVID as well, is reading lateral flow tests. So this could create, reading lateral flow tests can result in a lot of errors, simply just interpretation errors of reading the results of the lateral flow tests. So what this team in Africa did is collect many thousands of images of lateral flow tests and classify them using field experts, and then train machine learning algorithms to read the lateral flow test. So train the machine learning algorithms to read them like experts, so that patients or community healthcare workers can better interpret these tests. And in this study, they found that a class of machine learning algorithms known as convolutional neural networks can drastically improve the prediction accuracy, the classification accuracy of the lateral flow tests. Okay, so by just feeding it images of the lateral flow test result, it is able to classify whether the sample is positive or negative for HIV. And there was a huge amount of work collecting 11,000 lateral flow images. Another thing to note is that the algorithms were actually pre-trained on just ordinary images. So these are algorithms that have been trained on image databases like AlphaFold that have like ordinary images of different shapes and so forth. So they're already kind of learning from vast amounts of data to recognize different features from images. And this has spurred on a lot of advancement in kind of medical imaging. So for those who are like working on applying machine learning techniques to imaging or medical images, this is quite an exciting time, because these algorithms have been shown to be quite powerful, performing just as well as humans, if not better, because they can recognize, the faintest features, such as, faintest pixels in the lines of a lateral flow test. Now, one of the challenges of applying machine learning algorithms, especially in the clinical setting, is that we're trying a lot of different approaches, right? But when you're trying to, apply it to patients, you don't want to try a lot of different approaches. You want to kind of standardize a best practice approach, so that you can ensure equitable care to all patients. So a lot of work and a lot of the obstacles we need to overcome is trying to determine, out of these experimental pipelines that we might have published, which one would be best to roll out in a clinical setting. So I've just given a very quick overview of a few examples of a few applications of machine learning in drug development. This talented clinical fellow, Chen To, has written an excellent review article of other types of machine learning algorithms used in pharmacology, from drug design to patient stratification. So if you're looking for kind of a reference for different techniques, you can check out this review paper. But do note, it was 2 years ago, and this field is moving very fast. So there are much newer techniques that are available as well. So that's just a side note. And another side note is that we also spend a lot of time in helping non-computational researchers, like clinicians, to understand these data-driven approaches. And this is critical for uptake of machine learning algorithms in translational medicine and clinical medicine. And this involves really like running courses to teach them kind of simple data analysis techniques and building data visualizations, such as our Shiny apps to visualize the outputs of some of the machine learning techniques. Okay, thank you. I would like to finally thank my team, both in Sheffield and Singapore, and note that we're always looking for young scientists. And thank you to all the funders, and I would be happy to take any questions.